This research presentation is based on Hode's article titled Individual Differences in the Peripheral Immune System Promote Resilience versus Susceptibility to Social Stress. The research presented is based around depression and anxiety disorders. The National Institute of Mental Health describes depression and major depressive disorder as a serious mood disorder affecting how you think, feel, and perform daily tasks. The article describes depression and anxiety disorders as being associated with an increased release of peripheral cytokines, and differences in the sensitivity of the peripheral immune system can help predict vulnerability to social stress. In order to quantify individual differences, cytokine profiles were taken from various mice to find different levels of stress regulators. Two different types of stressors were used, RSDS, which is repeated social defeat stress, and witness defeat, which is a purely emotional stressor. This is because constant social subordination can lead to depression or depressive-like behavior. This behavior is described as social avoidance. For RSDS, aggressor mice were placed in a cage and experimental mice placed on a separate but clear half of the cage so that the mice could see each other. The mice were placed on the same side of the cage for 10 minutes each day for the duration of the experiment. The mice were moved to a new aggressor each day. The control mice were housed and moved in the same way. The witness mice were placed on one side of the cage and watched the aggressor and defeat mice, but had no physical contact with the aggressor mice, but were housed the same way as RSDS. The subthreshold witness defeat mice went through RSDS, then 2 weeks later were allowed social interaction. They were defeated 3 times in an hour to see how animals change when they become stress susceptible. This research hypothesized that pre-existing differences in the sensitivity of an individual's peripheral immune system dictate their subsequent vulnerability or resilience to social stress.
The main objective of the first round of tests was to determine if there was a correlation between the immune system molecule IL-6 and stress in mice and humans, and whether or not it could be predicted if a mouse would show the resilience phenotype to stress before a test was performed. After an RSDS test, it was found that in the mouse's blood there was an increase in IL-6. To clinically validate this observation, human testing was performed. 2 different pools of humans were polled, those with major depressive disorder, or MDD, and those who were deemed mentally healthy. Tests were ran and then observations recorded, which will be explained later in this presentation. A second test was performed with these pools of people. This test was used to determine if antidepressants acted on the immune system. Physical examinations were performed on the mice in RSDS to make sure the trauma did not affect social interaction. Testing was also done to determine whether or not it could be predicted if a mouse would show the resilience phenotype to a stress before a stressor was tested. Ex vivo samples of leukocytes were taken a few days before an RSDS stressor test and then after the test was complete to compare the makeup of each sample. The second tests were performed to examine the function of increased immune activation to stress. In this experiment, animals were reconstituted with bone marrow hematopoietic progenitors isolated from susceptible or control mice. Before injected with either of these 2 bone marrows, the host mice were irradiated with a lead helmet to ensure the central nervous system was unharmed, but the peripheral immune cells were destroyed to make room for the bone marrow of the donors. Once these mice were successfully irradiated, they were injected with one of the 2 bone marrows of the donor mice. After this, the mice were allowed 5 to 6 weeks for the donor cells to be accepted before performing the stress tests. It was also ensured that most microglial cells were not affected by radiation and that they were the host bone marrow cells before tests were performed. Stress tests were then ran and recorded. We will talk about those later in this presentation. This third test was aimed to determine whether alterations in peripheral IL-6 contributes to differences in susceptibility to RSDS. For this to be done, 2 different chimeras had to be constructed. The ones that were made were IL-6- and wild-type bone marrow. Different stress tests were then ran on these 2 different chimeras. After this was completed, IL-6 destroying antibodies, MAB, IgG, and saline, were administered to see if they had any effect on stress. These slides will begin the results part of our presentation. In slide 1, it is the human testing. After clinical testing was performed, it was determined that subjects with MDD had higher serum IL-6 than those with no depression. Depicted in figure 1C, the black line represents the subject with MDD. There is a significant difference in the amount of IL-6 in mentally healthy patients compared to the patients with MDD. The final 3 graphs on this slide show that standard antidepressants are not acting on immune function to exert their behavioral effects. Supporting evidence for this is shown in figures 1D and figures S3B and C. Figure 1D shows the IL-6 levels of mice that underwent 10 days of RSDS and then 35 days of imperamine treatment. And figures S3B and C show the still uplifted levels of IL-6 after these antidepressants were administered. In results slide 2, we depict the outcome of predicting resilience phenotype. After the RSDS test was performed on all of the mice, it was shown that the mice that had more leukocytes circulating 4 days before the RSDS test was run were the mice that showed the resilience phenotype, as shown in figure 2A. Figure S3E depicts mice that had lower circulating leukocytes that acted normal after the RSDS stress test. Slides 3 and 4 depict the results of the bone marrow test. After successful transfer of the donor bone marrow into the host, the stress-susceptible bone marrow chimeras overall had more circulating leukocytes than the wild-type control chimeras, as shown in figure 3B. Stress-susceptible bone marrow chimeras displayed increased social avoidance following subthreshold defeat compared with controls in figure 3D. There were no differences in the measurements of anxiety-related exploratory behavior indicated by the elevated plus maze. In table S2, you can easily tell there were no significant differences in the amount of time the mice stayed in the different parts of the maze. Slides 5 and 6 show the results of the tests ran with altered IL-6. After the development of the 2 different IL-6, a 10-day RSDS was performed on mice containing these 2 different molecules. After the 10 days, it was concluded that IL-6 minus bone marrow chimeras and IL-6 minus mice acted the same with similar levels of resilience measured by the increased social interaction ratio shown in figure 4b. Due to this, it is assumed that leukocyte-derived IL-6 is crucial to the development of social avoidance. Next, the mice were exposed to the same 10-day RSDS, but this time IL-6 minus and wild-type bone marrow chimeras were exposed to the witness model. Bone marrow chimeras from the high IL-6 releasing controls displayed robust social avoidance behavior, while the IL-6 minus bone marrow chimeras demonstrated resilient behavior shown in figure 4C. Finally, 3 different compounds that are used to neutralize IL-6 were tested to see if they would eliminate the response of social avoidance after a stressor. Pharmacological blockade with IL-6 MAB but not IgG or saline alone prevented the development of social avoidance shown in figure 4E. It is also important to note that MAB did not reduce anxiety-associated exploratory behavior in the elevated plus maze compared to IgG MAB.
This research showed that an increased risk of developing a stress disorder came with sensitivity in the peripheral immune system. The IL-6 levels were a good indicator for social stress behavior, and leukocyte levels could be an indicator of stress susceptibility. We would also include non-drug resistant mice in the study just for comparison. We thought that the non-drug resistant mice might have a difference in core body temperature versus the no difference that the study found between the control and the chimeras. We would also test female mice since their bodies may react differently to stress than the male mice. This study only used male mice since it is believed that the stress phenotypes are passed down paternally, but females can still express stress phenotypes. Because of this, we would want to test them so that any experimental drugs can be designed with their physiology in mind.